question 5 now. The RHD antigen is a protein found on red blood cells in humans. The presence of the protein is genetically controlled, and there's possible phenotypes and genotypes with respect to this trait, and they're shown in the table below. So we see that the genotype big D gives you rhesus positive, and it means that the RHD antigens are present on the cell. The same thing here, big D, little d, rhesus positive present, and we find that little d, or the recessive is rhesus negative, and the antigens are absent. So we clearly see that when we look at this, the ability to be negative is obviously recessive, and being dominant means that you're positive. With reference to the father's possible phenotypes and genotypes, so remember we've got to look at both, explain how a rhesus negative mother could be pregnant with a rhesus positive fetus. So if the mother, that means is little d, that means the rhesus positive fetus must be big D, or big D. And so what that means for the father's possible phenotypes and both genotypes is that the father must be rhesus positive. And that's the thing that we've established here, given that if the mother is rhesus negative, the father must be rhesus positive. So that's what I've actually mentioned in my first dot point. But what are the possible genotypes? And the possible genotypes are little d, so big D, or big D. It doesn't actually matter which one, because that will give rise if you perform the cross, which I'm not going to do now. But if you do perform the cross, if you obviously have big D, all are going to be rhesus positive, and if you have big D, you're going to have half, which are going to be rhesus positive, and half which are negative. And so that's why the possible genotypes are big D, or big D, which therefore makes the fetus heterozygous. So the fetus must be heterozygous if it's going to be a rhesus positive fetus, therefore big D. Okay, so moving into part B now, hemolytic disease of the newborn, HDN, can occur if a rhesus negative mother is pregnant with a rhesus positive fetus. So with the above situation that we had before, during pregnancy and birth, some fetal cells, may enter the mother's bloodstream, and therefore the mother makes antibodies, actively makes it, against the fetal RHD antigens. This is usually not a problem until there is a second pregnancy with a rhesus positive fetus. RHD antibodies made by the mother then cross the placenta, and that results in the possible death of the newborn. So B is explained by referring to the immune system, so we must refer to the immune system, why the fetus in a second pregnancy is at a far greater risk of HDN than the first rhesus positive fetus. And when we look at this, what I've actually constructed before is just a little bit of a diagram. You see P1 and P2, and this is just extracting the information of what they've told me from this question stem. And when we look at it, or the stimulus, and so pregnancy 1, the mother produces the antibodies, but there are no complications from this. In pregnancy 2, those antibodies actually cross the placenta, and due to the memory, there results in potential fetal death. And so what that shows me from this is that during the first pregnancy, there must be B memory cells that are produced, because we know when B memory cells are produced upon the second infection, if you will, or invasion, the second response occurs, will be larger and more rapid during that second pregnancy. And that is what may lead to the possible death of that newborn. So the 2 dot points I've got are very simple. During the first pregnancy, the B memory cells are produced. They must be produced because therefore in the second pregnancy, there would be a larger and more rapid response due to the presence of these B memory cells, and the antibodies that would occur, or that would be produced, would actually lead to that particular death during that second pregnancy. Okay, moving into part C now. There's a treatment called Immuno Prophylaxis. That's a big word. Has reduced the incidence of newborn death due to HDN. In this treatment, the rhesus negative mother receives injections of RHD antibodies with her first rhesus positive pregnancy, and then again at the birth. Would you consider this treatment to be active or passive form of immunotherapy? And then we have to explain our response. Now, the first thing I think of, has the mother actively produced her own antibodies? That's the big thing, because we know that's the resulting in active. If it's passive, it's obviously due to the presence of the injections. So the first thing I'm going to say is passive. You actually didn't get a mark for this particular statement of saying it was passive. But once you actually understood it was passive, why is it passive? Because there's no B memory cells that are produced by the mother. The mother isn't producing any of these memory cells because the antibodies are being injected into her system. And so that leads to the third point, and that's largely because the mother doesn't produce her own antibodies. They are delivered via injections. Again, understanding active versus passive, active being the mother actively produces these antibodies versus passive, whereby she's actually receiving these particular antibodies. And so when we really boil that down, it's no wonder why 19% of the state only got 2 out of 2 marks, and that actually left 39%, if I write it over here, that got 0. And the reason why I think that is that people would have got caught up in the detail and most definitely made contradictory statements within these particular lines. Remember, ask yourself, is it passive or active? Then, once you've decided it was passive, ask yourself, why is it passive? And it's passive because there are no B memory cells that are produced, and thereby she doesn't produce her own antibodies, and that leads to a passive form of immunotherapy.